Hello and good evening. Actually, welcome to Chapter 26, Phylogeny and the Tree of Life. I'll start up my timer right now. This is going to go a little bit longer. Basically, we're going to try and cover what is taxonomy, phylogeny, shared characteristics, and lastly, kind of building cladograms. Basically, through this, we're going to be looking at kind of how life is arranged. It will also kind of help you understand a little bit about the different forms of life. Again, these are all artificial constructs. These are all man-made. And again, we are trying to kind of match what's going on with life. Again, you look at this organism and you would say, snake. Is it really a snake? Nope. It's a lizard. Okay, there are certain characteristics that this organism has that make it look like a snake, but it's not a snake. And again, the reason why we do that is there are certain characteristics that this organism has that make it less snake-like. For the most part, we've talked about the three kingdoms. We've talked a little bit about some phylums and some classes and possibly even some orders. But I don't really kind of want to get into this because this is an artificial construct. And I see a lot of people more memorizing what a phyla is, what a class is, than kind of really understanding what the relationship is between these organisms and their differences and how we actually look at it. And so let's possibly throw this out. You need to know the three domains, and you'll need to know some phyla, but that's about it. Again, remember in hierarchical classification that in the Linnaean system, we use a binomial classification. That is the genus species. The genus is always capitalized, and the species is always lowercase. Again, it's always either italicized or underlined so that you could always see it. Again, we're looking at these kind of different organisms here. And they are all the same in that they are all mammals, and they are a part of mammals in that they are carnivora. Again, we have the cats, we have kind of the weasels, and then we have kind of the dog-like structures. And again, they're all related to one another. We think that they had a common ancestors, and they share characteristics the same. So what I wanted to get you to do is to think about how we organize things and how we understand them. Again, what we call phylogeny is the evolutionary histories of species or a group of species. That is, who is related to who, or who is most distantly related to who is most closely related to who. Again, this represents the hierarchical classification. It represents our dichotomous key and kind of how we tend to look at things. Again, when we look at a phylogenetic tree, these individuals that share a most recent ancestor are what we call sister taxa. Again, where two systems branch out or more is what we call a node. And then we have our ancestral lineage. Polytomy is one that has many sister taxa. You just don't have one or the other. And again, this is a representation of a hypothesis of the evolutionary relationships. And that's basically all that it's looking at. And so, again, in a phylogenic tree, we show patterns of descent, not phenotypic similarity. And so a lizard and a snake may look like one another, but they are not the same thing. In the past, when things looked that much alike, we would classify them as being alike. Tree branching does not necessarily indicate actual absolute ages of the species, just kind of relative ages. Again, evolution takes a little bit of time, and we think that things that are more related to one another shared a common ancestor a little bit closer ago. Sister taxa do not evolve from each other. They're more like cousins. They share grandparents. These grandparents may be long gone. Okay, they're similar to one another. Again, these are two phylogenetic trees that we look at. And one here, this represents kind of the age of when they went, and this is looking at kind of genetic changes. And so, again, Drosophila, we think, shared a common ancestor with human beings a reasonably long time ago. And then, again, we look at here in the genetic changes that there are many genetic changes between the that and us. So this is just another way to kind of look at it. You can put this on a timeline, and we'll talk about how those timelines are looked at. Again, phylogeny is important because, again, we're looking at similar characteristics closely related species. Why would we want to do this? Well, things like today, if you go to a grocery store, there are certain endangered whales, and for the most part, you can't tell the difference between whale meat. Actually, how many of you gone to the store and bought some white fish, and it was tilapia? How do you know it was tilapia? And basically, what they've done is they set up a phylogenetic tree using DNA PCR analysis, and I'll leave you kind of to look at that. And so this is a test that we can do, and maybe in the future you will have a test, something like this, where you could go to the store and you could say, hey, this is a specific type of meat. Okay. How can we build a phylogenetic tree? We have cladistics use similarities, shared characteristics between organisms to infer relationships. Now, this is kind of a hard one. Almost anything can be used to infer these relationships. I like DNA. You can use the bones in the jaw. You can use the shape of the teeth. An ancestral character is a character shared beyond the taxi we are trying to define. And so that would mean everyone in an area has that particular shared characteristics. And a shared derived character, evolutionary novelty to a particular clade. And so it's something that only a certain group is going to have. You guys are going to need to know what a monophyletic clade is. That's where an ancestor species and all its descendants. A paraphyletic grouping is anthro-species and some of its descendants. A polyphyletic grouping is various species that lack a common ancestor. And again, we're looking at here. And so these guys here lack a common ancestor. Again, we go back far enough, they have a common ancestor, but we don't have that here between those guys when we're looking at that particular grouping. And so again, we have a problem with the five kingdom system in that the Monera and the Protista are basically polyphyletic. So again, that doesn't work. It's one of the reasons why we got to cut it out. You know, again, shared derived characteristics are more likely to be found in taxa that are A, monophyletic, B, paraphyletic, and C, polyphyletic. Can we build a phylogenetic tree? Cladistics uses similarity, shared characteristics between organisms to infer these relationships. And then we already talked about shared ancestral characters and shared derived characters. It's the second time that we've talked about it. And so, again, let's take a look at some classic vertebrates, okay? And so if we take a look at these vertebrates, we would see that, again, a vertebral column, a hinged jaw, four walking legs, an amnion, hair, are all things that we can use to kind of separate these guys out together. And so basically what we do is we assume that each of these is important to another, and then we compare relationships. An outgroup is known to have diverged before the lineage we are studying. It's outside of that lineage of interest. And so, again, when we look at these vertebrates, these organisms all have jaws. Here's an organism without a jaw, and that would be an outgroup. If you're using cladistics to build a phylogenetic tree of cats, which group of the following would be the best outgroup? A lion, a wolf, domestic cat, a leopard, or a tiger. If you kind of know what these are, you would know that the wolf would be. Again, these are all carnivores, but the wolf is definitely not a cat. It doesn't have some of the cat-like characteristics that you would think about. Again, tricks for a biologist, you want to try and sort what we call homology from analogy. Again, a homology is a shared derived characteristics, whereas an analogy is something that's similar because, well, think about it. Okay, analogy: similar traits arose independently due to not due to ancestry, and so that would be on things like snakes and lizards that look alike. And so, if we go over here and we look at this, we would say that salamanders and lizards look alike, that the bird's an outlier, that eel and snake look alike. Again, this is not the case. When we really look at this, we look at homology, similar traits due to common ancestry. We see that lizards, snakes, and birds are all very similar to one another, that the salamander is a different group, and that the eel is a very older group than that. We looked at the legless condition, parsimony versus likelihood. Maximum parsimony is the least amount of evolutionary changes that have occurred. Maximum likelihood is the most likely sequence of evolutionary events that have occurred. And so things have a basic order to them, and a lot of times we need to kind of look at this kind of basic order. And we'll get to that in a little bit. Again, with as little as 50 species, we have roughly 3 times 10 to the 76 different possible trees. And so not all of them are as likely as others. And so we need to kind of look at that. I'm going to pop over in this. And so basically we could look at the differences between these sequences, comparison of possible trees. And again, we would say the tree that is the simplest is probably the most likely. And that is number 1 is more likely than number 2, because at number 2 we see a lot more change. I'm going to go over this. Again, so let's go back. When we look at life, there's 3 major domains in life: the eukarya, the bacteria, and the archaea. Again, there's a common ancestor that we may think may have been a little bit more like a bacteria than an archaea and a eukarya, but that organism is long, long gone. Some of its remnants are here, but that organism is definitely gone. What we kind of see, though, is that we have this process called endosymbiosis. And what I wanted to talk to you about, kind of get an idea for you, is that, you know, we tend to think of an organism having an offspring, and those offspring having their own offspring, etc. But what would happen if one species was to influence genetically another species? And actually, we've seen that a number of times. In this process that we call endosymbiosis, we see that the more complex cells, the eukaryotic organisms, came about because 2 relatively simpler cells came together and started living together and shared their genes. 2 totally separate organisms. Amazing. And I think the more we see this, the more we see it. And what we see in animal cells, mitochondria in our cells were once actually free-living, sugar-eating bacteria. In plant cells, they have mitochondria like our cells. They also have chloroplast. And we think that the mitochondria, again, endosymbiosis, may be a little bit older than the chloroplast. Evidence for symbiosis is very similar in that the mitochondria and chloroplast look like organisms. And what's really kind of interesting is they have their own DNA, they have their own special RNA, they have their own set of ribosomes, and they divide very much like the cells, just like real cells. Okay? And so again, I leave you to kind of look at this series a little bit, look at the series of slides that we have in there. Again, Darwin, in looking at his kind of universal tree of life. And so what Darwin did is he thought that there was an original ancestor, and then that ancestor begat the next one, which begat the next one, which possibly had a change, and then I'm drawing this upside down, so this is his actually original tree over here. But what we're seeing is that may not be the case, that we had this horizontal gene transfer. Is that horizontal gene transfer going on today? We are really situated to find out, and we may see something like that. And so if we really kind of look at it, this in this modern theory of life is that in this very early stage we had organisms swapping genes kind of indiscriminately, really kind of cool. I'm not going to get into parsimony of the birds here, and then we'll do several examples in class. Again, you need to know this vocabulary, and we'll go over it. So, and that's it. Thank you.